All right, good afternoon, everybody. This is Dr. Eric Toring. I'm the Senior Vice President for the American College of Veterinary Preventive Medicine, and I want to welcome you to the webinar this afternoon. Today's webinar is sponsored by the American College of Veterinary Preventive Medicine and its partner organizations, the American Association of Food Safety and Public Health Veterinarians, the National Association of Federal Veterinarians, the U.S. Animal Health Association, and the National Association of State Public Health Veterinarians. It is now my pleasure to introduce our speaker for today. Dr. Julie Lennick is a veterinary epidemiologist with the U.S. Department of Agriculture, APHIS Wildlife Services. She is currently serving as the National Coordinator of the Wildlife Disease Program. Dr. Lennick has been with APHIS for approximately 8 years after switching gears from emergency and trauma clinical medicine for 14 years. Dr. Lennick received her DVM from Colorado State University, a master's in public health from the Colorado School of Public Health, and is a boarded diplomate of the American College of Veterinary Preventive Medicine. So welcome, Dr. Lennick.
I'm going to go through slides, and I'm going to be covering today the current outbreak of high-path avian influenza in the United States, but from the wildlife perspective. I work for APHIS Wildlife Services, and this has certainly been a complicated and twisting and turning outbreak. And I'll go a little bit back in history and then kind of go forward as to, what's been going on the last couple of years and where we are right now. And I'll dabble into the connectivity and the sort of overlap of wildlife with the current situation on the domestic side. So I hope it will be educational to you. I'm already going to pre-apologize to any of my USDA colleagues who are out there because they've probably heard versions of this talk 50 times. So you can hang up right now. You don't need the CE credit anyway. All right. Perfect. Well, get started then. And thank you for the introduction. As Eric said, I'm with APHIS Wildlife Services. I also wanted to note that I serve you in the World Organization of Animal Health, WOA. I am the national focal point for wildlife for the United States. And what that means is Dr. Rosemary Sifford, who is our Chief Veterinary Officer for the U.S., has designated focal points in several areas of expertise or, as a point of contact, essentially. And I am the wildlife focal point for the U.S. So if you ever need anything in or out of WOA, I'm your gal for that also. Okay, let's get started. Okay, I'm going to go into kind of multiple data streams because this current outbreak is complicated and has been convoluted and is keeping us on our toes. And I'll kind of explain what I'm talking about, hopefully, and then how all of these data streams merge together. So we look at multiple methods of surveillance for avian influenza. Kind of our main bread and butter is our targeted surveillance of dabbling ducks. These are migratory waterfowl. That's like the regular source for avian influenza. We are now looking at morbidity and mortality investigations because this particular avian influenza has caused vast sickness and disease in our wild bird populations. I'll talk a little bit about two special projects that are going on this year, which is our spring enhanced surveillance and our targeted surveillance around livestock facilities. So, and all of these kind of 4 different chains of information merged together to give us our information and our intelligence in wildlife for the outbreak. Okay, so the routine regular wild bird surveillance. This is looking at apparently healthy birds. Our method of capture for this is, or our method for sampling is live capture in the summertime. Birds are literally caught live. That's done in conjunction a lot of times with State Department of Natural Resources for banding. So these birds have Fish and Wildlife Service bands on them so we can see where they moved or if they're harvested. And then they're sampled, then we let them go and they continue to fly wherever they were planning on going. In the fall and winter, we rely on hunter harvest. So those are duck hunters. And we ask for voluntary participation from duck hunters to swab their birds. And sometimes we look at agency collection. So we have some airports where birds are removed to reduce the threat of bird strike, for example. And if they're removed for those purposes and they're within our sampling regimen, we sample those as well. So kind of whatever we can get our hands on for dabbling ducks, those are the natural reservoirs for avian influenza historically and in this outbreak as well. And they have the highest prevalence. So we can get the most information from the least amount of birds sampled. And that kind of gives us our surveillance, which allows us to understand prevalence, look for early detection for new viruses, understand new areas of risk, and look for introductions. So this system has been in effect for some semblance since about 2005. We're almost 20 years into this, and it worked as it was supposed to. We did detect the first H5N1 in the United States in our wild bird surveillance from samples collected in North and South Carolina all the way back in December of 2021. So unfortunately, we are approaching our 3-year stint into this current outbreak, and I'll kind of talk about how this is ongoing and how this is different from what we have seen in the past. I'm going to just say HPAI or H5, but if you're a virologist and you're cringing, I will clarify right now that this that we are talking about is a Eurasian goose guangdong lineage. It's called H5N1 2344B. This specific one has been circulating in Europe since the year 2020. So Europe and then Asia are about 12 to 18 months ahead of us in this outbreak. In our North American detections, this was actually detected on the Atlantic coast of Canada in November of 2021 in the Newfoundland and Labrador province in a gull. And then very shortly after, about a month later, we picked it up again on the Atlantic coast in the Carolinas in these beautiful duck species. Okay, I'm going to take us backwards a little bit in time and kind of explaining the migratory nature of these waterfowl. These are birds that migrate thousands of kilometers on an annual basis. So the general gist is that the birds go north for breeding. They spend our American summer, May, June, July, August at the summer breeding grounds, begin a fall migration, migrate down through September, October, November, and then December through the spring, they are down in their winter grounds. So the map on the left kind of gives the example from the Asia Pacific side. These birds would migrate down here for the winter, stay there, come springtime, move forward, and then in the summertime, go to the summer breeding grounds. Our birds do the same thing, and they can coalesce in some of these areas. Kind of our Alaska, Russia area is an introduction. And then come fall, our birds will return back down our Pacific coast, which is a method of introduction, which is exactly what happened in 2014, 2015. The strains that were introduced and kind of mixed up here kind of came down our coast and started our 2014, 2015 outbreak. Zooming out even farther, these are the global migratory pathways. So now the Americas is on the far right. On our far eastern side is our Atlantic coast. Our migratory pathways overlap with the East Atlantic, which is the western coast of Europe. And these birds overlap in Greenland, Iceland, kind of the eastern coast of Canada, and then return their spread down during their fall migration. So that would be an introduction from Europe, would come down our Atlantic coast, in contrast to what we saw previously with an introduction or movement from Australia or the East Asian side coming down our Pacific coast, because they kind of overlap here in the Alaskan and Canadian area. So we look at our coastal areas as potential pickups for new introductions. And then we see movement within the Americas as the birds go up and down seasonally. The birds, of course, don't go straight north and south. And this is a paper we published a couple of years ago looking at banding data, those bands that I talked about. So we picked up animals that are birds that had been in the Carolinas and looked at like where the bands were recovered. So a lot of them stayed kind of on the Atlantic side, but even birds from the Carolina can make it all the way over to Montana and kind of the central and northern parts of Canada. So it's not quite linear, but we can kind of see these like sun ray effects for bird movement. So when we try to forecast where the disease may be moving from or introduced to, we kind of think about all of these patterns overlapping each other. Okay. So this is sort of the behind the curtain look as to what my shop does. Our team at the National Wildlife Disease Program designs based on a model, a surveillance plan year after year with 3 seasons. So we start in the summertime after breeding. And again, that's usually in earnest, our sampling starts in July after new birds are born. We have a summer season that goes to about Labor Day. The fall migration picking up September to December, and then our winter and spring season kind of merges into one from January to April. And then we leave the birds alone during their breeding and reproductive time to allow for excessive breed, and then the cycle starts again. So we start our year every summer following the biological cycle of the birds. For the current year that we're in, we'll be targeting about 38,000 samples from the 4 flyways. So this is the flyways sort of in different colors. Atlantic is on the far eastern side, the Mississippi flyway, the central, or we would kind of look at that as the plains or Rocky Mountains, and then the Pacific flyway, of course, on the western side. And we've extended this to Canada and Mexico because that's how big our bird migration is for the North American flyways. So our sampling contains all 49 states that are given a quota, a target number of birds to sample. In addition, we have opportunistic sampling from Hawaii and the Caribbean, U.S. territories of Puerto Rico and the U.S. Virgin Islands. And those are pulled out because we do not have avian influenza detections in those 3. We have detections in the 49 states, the 48 contiguous, and Alaska. We have not picked up high-path avian influenza in Hawaii, Puerto Rico, or the U.S. Virgin Islands. And that can be just explained by some geographic separation and maybe some protection based on their distance from the mainland. Our cycle begins every summer, and we use our targets, waterfowl populations, and detections to kind of change that year by year to try to maximize it and get the most information out of our surveillance. This is just looking at that same information, but in our watersheds. That's where migratory waterfowl live. They don't really live in state by state boundaries. They live in their own watersheds. So in the summertime, we were aiming for just about 8,000 samples. You can see a northern bias there. Again, that's where the birds are. The lighter shade of the shapes are a lower sampling number. And as the color gets darker, as the gradient gets darker, that's a higher sampling quota that's been given to that biologist or that state team. In the fall, you can see we're pretty well geographically distributed across the country. And then going into winter, we're moving down south. And again, this follows the migratory pattern and the movement of the birds. So altogether, those numbers add up to about 38,000 and change. And this is where we are right now. So as of, I think, earlier this week, our teams had collected just more than 12,000 samples. So we've completed summer and we're into our fall sampling. We're about a third done with the year and have representation, pretty nice representation, which is what we want. We want to have like a good snapshot in time, season over season across the geography to understand where the disease is not and what might be changing in the virus. All right, that was all regular surveillance. I'm switching gears now to talk about morbidity and mortality investigations. And I said earlier that migratory waterfowl are the natural reservoir. Those birds typically and historically carry the virus, but they have no signs of disease. You can't tell that they're sick. We really don't have any indication that they're infected. In the past, we really didn't appreciate much illness in wild bird populations. That is dramatically different in this current, I'm going to call it the 2022 outbreak. That's when it really started in earnest in the United States. But this global outbreak from 2020 onward has caused mass mortality in wild birds of so many different genuses and species and in so many different areas of the world. So we've seen a change in the virus compared to our previous outbreaks. We had a high path outbreak in the US in 2002 and in 2015. This time it's very different. It is making wild birds very sick, huge mortality events in the United States and globally. Many of these birds are showing neurologic signs, swimming in circles, head tilt tremors, seizures of sorts, or acute and massive die-offs that have gone on. I will explain, and I'll try to give the nuances as we talk about the reporting. When we talk about mortality investigations, we typically limit these to only a couple of samples in a large outbreak. Mortality events vary a little bit by state, but usually it's defined as more than 30 birds in a specific geographic area. We have awareness of some of these in the hundreds or even the thousands of birds that have died in some of our cranes and pelicans and goose populations. And we'll ask folks to sample 5. So the reported amount of mortality, I think, is a vast underestimate of the true scale of the mortality and the scope of death that has happened in wild bird populations worldwide. The mortality investigations in general are managed by the state. We don't have a federal coordinated plan for this. Wildlife in general are managed by the state agencies, your state department of Fish and Wildlife, or Parks and Wildlife, or Natural Resources. Our disease biologists go out on occasion. A lot of these have been picked up by wildlife rehabilitation facilities or raptor centers or those types of things, and then a lot of our university partners. So for mortality investigations alone, we've reported 5,600 detections since February. Those have occurred and have been reported in 49 states and in Washington, D.C., and they represent more than half of our detections. So again, previously our knowledge came from our surveillance. This time we're really fusing together both morbidity and mortality investigation information and our surveillance data to give us this big breadth of knowledge. And unfortunately, the mortality investigations continue to come in. This does not seem to be letting up in our wild bird populations. This is a breakdown just by very large bird class of what we're looking at. So the highest number are geese, swans, and ducks. And again, the 2,200 are confirmed detections. The scale of the mortality events is logarithmic scales above that. Vultures and raptors have taken a pretty heavy hit on this one. And then you can kind of see as it goes down into some of our shorebirds and some crane species. Owls of several different genus and species have been affected as well. Okay, this one always piques people's interest and certainly a sensitive topic. Our endangered California condors were also victims of this. The first detections of H5N1 in the California condor population came in March of 2023. We had a total of 22 known mortalities. And I should say that if you don't follow this as closely as some of us in the wildlife world do, California condors were on the brink of extinction. There's been a multi-decade project, the California Condor Recovery Program, for I want to say like 40 some years. The population is now up to 530-ish in the entire world. So 22 deaths in a very short period of time was certainly very concerning. Due to this, the United States Fish and Wildlife Service requested approval for a high-path avian influenza vaccine to be used in the United States for the first time in wild birds. We didn't have any safety or efficacy information on this, but felt like we really needed to do something because the virus had made its way into this endangered species. We did a quick safety trial that spring, April to May of 2023, and captured 20 black vultures and 8 control. And my Teams is beeping. Which are closely related. We vaccinated those birds and did not appreciate any vaccine site reaction or mortality, so felt that it was probably safe. and then move forward to do some check the efficacy, and then move forward with the actual vaccination of the condors themselves. We certainly didn't want to vaccinate and cause any illness in an already endangered bird. And then began the California condor vaccination trial. We had 10 birds in a group that were given 2 vaccines 3 weeks apart. We had a second group of birds who were given 1 vaccine, and then we watched their titers and have kind of watched these birds over time. It's been more than a year now for some of them. So 94 birds have, as of February, have been vaccinated with HPAI, and the condor mortality event seems to have gone away. So successful, caretaking and immunity looks like it's showing up for the condors, and at least for the time being, that mortality event has subsided, and we're keeping an eye on that population. Okay, this is where we are kind of all together. So the avian influenza in the United States, again, the colors in the background, the blue shades are our flyways. The gray shapes on top are our watersheds, and the red dots indicate detections in wild birds. And this is now more recent. This is since May of 2023 to current. So again, we're approaching almost 3 years into this outbreak. This is the most recent 15 months or so of data. So we are still getting high path detections across all 4 flyways and pretty geographically spread within the US. This is the total number of detections since the beginning of the outbreak. So again, we're north of 10,000. Again, that is an undercount of the mortality events, but you can see that this used to be a pretty map when we only had some of the states, but it's been stuck at 49 states for a really long time. Again, no detections of high path avian influenza in Hawaii, but every other state has reported detections in 2022, 23, and in 24. Same flyway colors in the background, but now I'm switching to detections of this high path avian influenza in mammals. The mammal outbreak and the detections started a little bit later. So May of 2022 was our first detection in mammals. And this has continued to kind of march on in a slow and steady way. We're unfortunately kind of running out of symbols on the right-hand side, but we have had detections in wild felids, wild canids, those are coyotes and foxes. We've seen it in mesocarnivore species like skunks and raccoons. We have reported it in domestic cats. Those are the black squares that you can kind of see throughout, a lot of them in the Rocky Mountain or in the central area. It has also spilled over into marine mammals, including gray seals, harbor seals, and a single bottlenose dolphin. So those are, of course, on the coastal areas. We can see those in Maine, Florida, and Washington State. And then kind of a smattering of other species, including mice, squirrels, some other rodents, and kind of a variety. Most of them are mesocarnivores, meaning they are animals that would be likely to eat a sick or dead bird, or kind of take an opportunistic free lunch. So the mammal spillover, and these are wild mammals, and I'm only saying that because we're separating them from livestock, which I'll get to in a tiny bit, are an interesting and I would say of concern, because anytime we have mammalian spillover, there's concern that this virus could be getting somewhere close to a pandemic potential virus. So the mammal sampling is handled the exact same way. They're sent from the exterior screening laboratories to the National Veterinary Service Laboratory, where they are sequenced and moved on. And I'll kind of go into that workflow, but these detections and the sequences are shared with all of our public health partners and kind of monitored on a continuous basis to watch for changes in the mammalian detections. Okay, so all of that pulled together, the H5N1 in wildlife, more than 10,000 detections in wild birds. That includes both surveillance, healthy bird sampling, and morbidity and mortality sampling from 174 different wild bird species and 399 mammal detections as of last week in at least 26 different species of mammals. So again, this disease, this current outbreak and virus is behaving much differently than what we've appreciated in the past. Many partners are working together on the wildlife side, on the One Health side, on our laboratory side, trying to investigate these sick or dead birds or animals and get them reported and investigated as soon as possible. All right. What we're seeing in the United States is not unique. It's not unique at all. Again, the introduction into the North America was on our Atlantic flyway, the fall of 2021. We saw pretty widespread movement through all of the Canadian provinces, and again, our 49 contiguous states, and then into Mexico. And then in this outbreak, we've then seen downward spread. So through Central America, reports of detections in wild birds, commercial and non-commercial or backyard birds, wild bird reports, and then mammalian and some human infections. So the disease has made its way through Central America, down into South America more than a year ago, moving into Colombia, Argentina, Brazil. Brazil is a huge producer of poultry, and they've been on everyone's radar for food supply for the whole world. And then, unfortunately, in our wild birds, it's continued to spread all the way down to Tierra del Fuego, or the southern tip of Chile and Argentina. The first detections have been reported in the last year in wildlife populations in the north part of Antarctica. Of course, some very special and usually isolated wildlife populations there, and in the Galapagos Islands. So the disturbing trends, mortality events, marine mammal die-offs, wild bird die-offs, and spillover between poultry and wild birds has continued globally. Again, this is just the Americas, but same reports coming in from Europe, Asia, and Africa. To date, Australia has been spared and has done a good job with their biosecurity, but they're pretty geographically isolated. Okay. I'll give you a little snippet about HPAI in dairy cattle. So again, this same H5N1 2344B this spring in March of 2024 was detected in dairy cattle. That first detection was in Texas, and kind of then quickly in sort of that Texas Panhandle, Kansas, New Mexico area. It has now unfortunately spread to 14 states that we are aware of. There are ongoing efforts from both the state departments of agriculture and our colleagues at veterinary services to control this with interstate movement and pre-movement testing. This is a specific genotype, and it's called genotype B3.13, affectionately known as the moo flu. The initial transmission into cattle is unknown, but we did pick up kind of the precursor, the most closely related to B3.13 in 3 wild birds and a skunk prior to the March event. And we believe the actual transmission or spillover introduction probably happened actually in December or January, simmered for a couple of months until it was detected with this mastitis and decrease in milk production in dairy cattle. And now we have subsequent cattle to cattle lateral transmission occurring through multiple states. So this is kind of our complicated version, and Richard Webby published a version of this first about 10 years ago, showing kind of wild birds as the epicenter. They're the natural carriers of it. We know that there is a risk from wild birds to domestic poultry. That's the concern always, is HPAI is going to get into our commercial poultry. In this outbreak, we've seen a handful of human detections. We have seen spillover and movement, back and forth between wild birds into the poultry. And then we've seen this just kind of bizarre and ongoing twists and turns of wild bird mortality events, changes in movement into mammals, including our mesocarnivores. These are skunks, fox, cats, the marine mammals, and then now into cattle. I'll talk for a second about pigs, but we do not have any detections of high-path avian influenza in swine, neither domestic swine nor feral swine in the U.S. That's always a concern because swine are a great influenza mixing vessel. So everyone wants to keep an eye on the swine population in this outbreak. Okay. If you remember when I talked about our kind of strategy and our sampling, We tend to have hands off of our birds in April, May, June to their breeding cycle. This, of course, was exactly when the first detection in dairy cattle came up. So we were given a task to take any samples that were possible to answer the question, is B3.13, the cattle strain, circulating widely in wild birds? So we launched a spring enhanced surveillance, which went from April to June. We asked our teams in the field to grab any opportunistic sampling they could, whether that was agency, harvest, or removal, any morbidity and mortality investigations in their states, or any live birds they could get their hands on. We asked for a maximum of 300 samples per state, and basically gave any bird species. And with this effort, we only detected high-path avian influenza H5 detections 2 times in goal species on the Pacific coast. So this is what that effort summed up to be. Our teams were able to go out and get another 5,282 samples. Again, great geographic distribution across the 4 flyways, excellent sampling coverage, and again, only 2 detections. So this kind of quick and dirty 3-month project gave us the information that indeed B3.13 was not circulating in our wild bird populations. And also we couldn't really find regular high-path avian influenza in these species either. But again, we went off of our normal sampling quota of our migratory waterfowl. So this gave us some assurance that the event that had happened and the transmission into cattle was not circulating widely around in our wild birds, and that we were going to flip back into our regular wild bird surveillance. And then we went to focus on following the detections in cattle. So B3.13, the cattle genotype, has now been detected and reported in more than 200 cattle facilities and more than 40 adjacent or kind of closely adjacent poultry facilities. So the same virus that went into cattle has now spread into poultry and is causing fatalities in 14 different states. Wildlife Services is following those detections, and we've been we've been in 7 states. The ones that are colored, again, our flyway colors are in the back. The colors don't mean anything as far as gradient, but we will get on these properties as soon as we are able to. After detection, poultry are stamped out, and then we will still go and visit if we can and get some sampling efforts to understand if there is spillover or residual virus getting into the wildlife after the detections of this virus in our livestock facilities. So we have been on 28 different farms, again, in 7 states. 20 of those were dairy facilities and 8 were poultry. We have been able to collect almost 5,000 samples from those efforts. So that's our peri-domestic kind of focused sampling. And we looked at 103 different species that live in or around those farms. And we find it. So this is the summary of our first round. We've just completed our first round of sampling in California. California, unfortunately, has dairy positive farms at this point. And in our different species, we have found it about 5.2% of the time, although we see very big variation in states. And I think that is a component of time from when the farms are positive to when we get some wildlife sampling happening. So we do find B3.13 when we do focus sampling on the farms right after the facility. So these are spill back. These animals are being infected from the dairy and not the source of introduction. Excuse me. Just to give an example of the sequences that we've done. Sorry, these go forward to the NBSL for sequence confirmation, and we are able to both get H5 detections from PCR and then use our whole genome sequencing to determine whether these are B3.13 in the peri-domestic species that are sampled from these farms. Okay. The swine question, because of swine's inherent ability to be great influenza mixers, we have ongoing questions. Could B3.13 or any other H5s make our way into our domestic swine population? The answer to that is not detected to date. And we've recently decided and started looking at H5N1 in feral swine. We kill feral swine all the time. They're an invasive species that are terrible in the Southeast, especially in the United States. My sister program, Dr. Dana Cole, runs the National Feral Swine Program within Wildlife Services. And we have been collecting samples for other diseases. And we've recently added on H5N1 as our screening. Of the 4,000 samples that we've screened for antibodies, this is just antibody testing for feral swine, 7 have been positive for H5. And we can't do any further subtyping to determine whether that's H5N1, but we just have H5 antibodies. We have recently, about 1 month ago, started doing nasal swabs on feral swine. Our first 12 samples have just come in and tests are pending. So to date, no detections of H5N1 in feral swine, but we are looking both on active infection with swabs and taking serum samples for antibodies. So we are looking for H5N1 in our feral swine population as well. All of these data streams pulled together are reported publicly in the United States and to the World Organization of Animal Health. Again, I'm our wildlife focal point. Our team maintains 2 pages, high path detections in wild birds. Again, that's just broken, unfortunately, the 10,000 mark on detections, and HPAI detections in mammals. Those are separate from 2 other reports on the domestic side, which have our domestic birds and our livestock, and those are maintained by my colleagues at APHIS Veterinary Services. So all of the information is out there and it's public. It just kind of has to be stitched together because of our different reporting lines. We've talked about this, kind of continuing the surveillance program, understanding spread, improving the science, and really being able to understanding any viral changes that are going on over time. I'll take a second to address the human detections. We do in the United States have workers who have been detected with H5N1. We have no fatalities in the United States. Most of these workers have been in very close proximity to dairy or poultry farms, mild symptoms, conjunctivitis, mild flu-like. And in this year, in 2024, we've had reports coming in from Colorado, Michigan, and Texas. And in 2022, we had 1 isolated, again, a close contact in Colorado. These are handled with our Department of Public Health and our human health colleagues, and of course, the Center for Disease Control and Prevention. CDC continues to consider this low risk to the general public. We do advise that our poultry and dairy workers have a higher risk and are working on personal protective equipment and some other routes to break that exposure. Guidance for humans, and again, these are folks that are in close contact with known infected animals, proper protection, appropriate personal protective, including gloves, boots, coveralls, eye protection, and respirators. And then wildlife services, because of this known risk, wild birds across the country are known to have high path. We've been coming up with a series of biosecurity, trying to break this transmission cycle from wild birds into our poultry or backyard facilities. We'll do biosecurity assessments, physically going out and doing an assessment and looking at wildlife in areas where there may be possibilities for contact. We also work with feed mills and zoos and some other captive animal facilities. Again, because it's somewhat ubiquitous in our wild birds, we want to try to break all those points of transmission. We inspect facilities, plug holes, put barriers or other nettings and deterrents up, advise our producers to make sure they're disposing of waste, including compost and carcass piles, decrease scavengers, decrease access to food or water. Wild birds are lazy and they'll just stop where there's a great place for lunch or they can rest. Getting rid of plants and things that are enticing. So any place where migratory waterfowl, that's a great landing spot. If I was a mallard and I was on my way, I'd stop in that field and just hang out for a couple of days. So we want to avoid any of those possibilities for transmissions because we know the wild birds are going to keep continuing to migrate and that many of them may be harboring us. All right, so here's my last couple summary slides. So this is a weekly summary of high path detections in wild birds. We're rolling back to January of 2022, so you can kind of see some seasonal trends here. we have our first introduction and peak where things were very busy in the summer of 22, slowed down, kind of quiet in the summer of 22, large peaks in our first fall migration. This is the fall of 22, and then things kind of quieted down. Very quiet for the summer of 23. In general, this virus seems to simmer down when it's very hot. And then we got a return in detections. This is the fall of 23, kind of quiet, and then some sort of peaks and valleys as we've gone through the summertime, still getting some spikes in detections going on here. Again, this information is cumulative, so includes our targeted surveillance around those poultry facilities, our spring enhanced surveillance, and our inclusion of morbidity and mortality investigations. So we do appreciate fall peaks. This is kind of the same information in a month by month. So these are our apparent prevalence is the line. So this is the number of samples that we collected versus the number that were positive. Right around here, we're running at like 6 or 7% of the apparently healthy wild birds. It went down the summer of 22, up to almost 15% of the birds that we were sampling in fall of 22, quiet down in the summer of 23, another peak in fall of October of 23, and then kind of bouncing around in 24. The bars on the back represent the amount of samples that we've taken. So despite sampling a pretty good amount, this is almost 1,500, 2,000 samples a month, we haven't found it this summer. And I will not use a crystal ball to predict what will happen in the fall of 24, but we're certainly ramping up our sampling efforts, and we'll be reporting that out if we start to get more detections in our apparently healthy birds. Okay. Kind of big picture summary is that H5N1 continues to circulate in our wild birds. We typically expect a surge associated with the fall migration. New birds were just born. They don't have any immunity. They will begin the migration right about now and travel all the way through the Americas, some down into South or Central America. The cattle genotype, B3.13, has been detected with targeted surveillance in and around those infected livestock or poultry premises. And B3.13 has not been detected in free migratory waterfowl. So we believe that the cases that we are getting are spilled back from our livestock situation that's going on, and not the source of introduction from cattle to cattle. I'm going to skip this. This is all the diagnostics, but I did want to show you a pretty picture that our colleagues at the National Veterinary Services Laboratory, that's the big laboratory in Ames, they do genome sequencing, and they have separated this out into multiple different genotypes. The very top line are the poultry genotypes over time through the outbreak. The middle line are the mammalian genotypes, and then our work that we're working on are the wild bird genotypes. And just kind of ebbs and flows and introductions of as the virus makes tiny changes over time, and we see some kind of beautiful patterns. What we see in the wild birds gets into the poultry and then gets into the mammals, and kind of the same information over the same time. The wild birds are in the back; those look like mountain scenes, and then the bar graphs are detections with the poultry premises overlaid on the top. So we will continue to build this body of knowledge together, tying in wild bird mortality information, our wild bird surveillance, any information and intelligence that comes from the poultry side, and then now, unfortunately, any information and intelligence coming from the dairy side. So a big One Health approach to this, of course, there's our standard One Health plug, but this really is all hands on deck from wildlife, agriculture, human health, our food safety, everybody working together to try to stitch this outbreak together and share information as rapidly as possible. I would be remiss to say, even on the wildlife side, we don't do this alone. We have an interagency steering committee with the United States Department of Agriculture, our colleagues in the Department of Interior, including the USGS, the U.S. Fish and Wildlife Services, and the National Wildlife Refuge. And then we are partnered in that with the Center for Disease Control and Prevention. And that group works together and kind of think tanks together every week to see if we need to make any adjustments or share any pertinent information with each other. There's my team, always amazing people doing a fantastic job. And I will stop there. That is me. I was privileged enough to go up with our Alaska team last summer and work on the Alaska project, which is as remote and far away as you can imagine. So I will stop there and happily start to look and take some questions. All right. So someone asked why there were no samples from Washington State during the enhanced spring sampling. Honestly, we didn't have a disease biologist at the time, and they were busy on a couple of other projects. The Washington State project. This was honestly a quick and dirty opportunity, and we just wanted geographic spread. We didn't have every state like have a mandatory quota, and it was what people were able to do in combination with their other projects. So the Washington State team honestly just may have been occupied with other wildlife projects. And I guess I was very remiss to say I'm talking about high path avian influenza. Our team on the disease program is responsible for wildlife mortality and sampling protocols for all diseases in wildlife that have threat to agriculture. So we are preparing for African swine fever. We're working on a hantavirus investigation. We do operational activities for chronic wasting disease. Our folks are working on tuberculosis and bovine TB. So we're not a one-trick pony, and we're juggling all of these things all the time. High path avian influenza is kind of the national emergency right now, but we have other duties to do. And in some cases, our teams are just stretched too thin to be everywhere all the time. So just no opportunities from Washington State. There wasn't a specific omission there. Someone pointed out that maybe there seems to be a bias in the wild mammals compared to other states. And early on, and I wonder if I can pull back up an older one, but we certainly actually, it looked for a long time, we had this like magic I-80 line across the United States, and all of our mammal detections were coming from the northern states. I will hypothesize a couple of things and then say I'm not totally sure. The virus seems less environmentally stable in hot, warm weather. So the South, maybe the virus doesn't last as long in sick or dead birds. Could be prolonged a little bit more in the northern area. So if you're a scavenger or a mesocarnivore, skunk, raccoon, fox, you might have more opportunities and more virus available in the environment in the north, where in general, it might be a little bit cooler year round. I will also say that we have some states that have very well-known wildlife rehabilitation programs and really good connectivity between wildlife rehab universities and their state departments of wildlife. Minnesota has a stellar program. Michigan has a great program, and they might have simply been more aggressive at looking for things. The mammalian detections in general have been in clinically affected animals, neurologic as well. So we've seen seizures, coma, tremors. Some, at the beginning at least, it seemed like younger animals, juveniles, were more likely, and maybe that was like a dose-dependent thing where if a fox brought it home to her kits, the kits maybe got a higher viral load per body mass or a lower immunity just in general for being young, and then they were clinically affected and died. And I think some states were simply more aggressive at working those up. Lots of the mammals came in through kind of interesting pipelines where they were ruled out for other neurologic diseases, rabies and distemper being high on the list. And then many of them we kind of found later. So once we realized, oh, foxes could have it, skunks could have it, coyotes could have it, people went back and rescreened samples that had been negative for rabies and then turned out positive for high path avian influenza. So I think we may have a state bias or some different capabilities or interests, and the northern states really jumped on it pretty aggressively. And if I'm not answering these, we can circle back and if anyone has any follow-up questions. But the next one says, does the USDA rely on states to report morbidity and mortality in wild birds, or do we all contact the agencies to make sure? It's a combination of both. In the United States, wildlife are managed by their state agencies or the tribe that they live on. So state and tribal autonomy and regulatory authority over the wildlife in their state. So it would be up to the state of Wyoming to investigate morbidity or mortality investigation in Wyoming. However, we've done this in a very partnership and collaborative way because we have a national and international responsibility for reporting high-path avian influenza. So it's handled initially at the state level. We do bounce these back and forth. We get reports and send folks out or vice versa. And we certainly at the federal level have supplemented and assisted states who can't get there fast enough or it is exceeding their capability to do that work. So state level for morbidity mortality investigations, but that information and intelligence is shared. Our partners at the Department of Interior, that's Bureau of Land Management, U.S. Fish and Wildlife Service, National Park Service, they would be handling the investigations on Department of Interior land. So federal partners have certainly been key in this, as have our tribal partners reporting any die-offs or mortalities on tribal lands. Any comments on why the pathogenicity remains high despite genotype changes over time? Lovely question. I don't know the answer. This 2344B clade that is circulating worldwide remains pathogenic. We do think we may be seeing some building immunity in our mature wild bird populations, but again, our migratory waterfowl, some of them are short-lived. We've got an annual cycle of new and juvenile animals that come in, of course, every year and then don't have any immune status. And we kind of start the cycle over again. So I don't know. I would love for it to simmer down or to start to be either less viremic over time or a different strain or genotype comes in and is a better fit. But this particular H5.1 seems pretty sticky. It's a good fit on our wild birds, which is why it has circulated around the globe. Do you think that wildlife surveillance around the infected dairy is adequate to monitor for changes in the dairy cattle? So I think there's a couple of components there. So the dairy cattle are being investigated by our state animal health officials. There is mandatory pre-movement testing for interstate movements. And those are all being handled on the veterinary services side. Those are also sequenced. We are looking at areas where we get permission to go on farm and do these like targeted sort of strikes to see if it's moving beyond that. So if there is discarded milk or manure or other environmental contamination from the dairy cattle, is that virus sticking around that farm for a while? And what is the exposure to the peridomestic birds? Those are starlings and grackles and sparrows and all the little birds that love to live in the eaves of barns. And then any mammals that may be visiting, raccoons, skunks, stray cats, coyotes, those kind of things. So I think it gives us a picture of what might be going on. And it is an indication, of course, with sequencing, we can understand if we're picking up any mammalian adaptation or any other changes there. This B3.3 seems pretty stable. It's in the dairy cattle, it's sitting there, and we are not finding it circulating out in our migratory wild birds. So I think stay tuned to that. I know it's September, so we're, I guess, roughly 6 months into the dairy event, but it's a fluid situation and we're learning as we go. And that event is certainly something to keep our eye on. And boy, we've got all hands on deck in multiple different ways for that. Is the surveillance in wild birds sufficient to detect spillback from cattle? Yeah, I think so. Again, so last year, our wild bird surveillance was about 42,000 samples, apparently healthy birds. There's thousands more that are submitted through morbidity mortality investigation. And then through our spring enhanced surveillance, the targeted surveillance around the farms, and then our regular wild bird surveillance, even since April of this year, we've collected 22,000 samples. I have pretty high confidence that if we were seeing it out in the wild, we would have picked it up, and that we are watching very closely for any genotype or reassortance or changes over time. And we'll continue to do both our regular wild bird surveillance and then work with our states on morbidity and mortality investigations. So yeah, I think we've got good confidence in what we're seeing right now. Again, the virus kind of keeps taking us on these little bits of twists and turns. So we will remain vigilant and we'll report any changes as if that's pertinent and important information. Can I go back to the slide of the testing results with the proximity? Oh, yeah. Okay. Hold tight. Let me, that is asking if I can share and I think maybe explain which are H5s and which are subtypes. So this is the geographic distribution of our sampling, farms, and then the number of samples taken per state. These are the sample totals based on the prems. So 28 premises in the States. We still have one team on the ground in California right now. I think this is what they were asking about. So what is the difference between our prevalence and our H5 detections? So the H5 is a PCR result. So we do first a PCR screening for influenza A. If those are positive, they are further screened for H5 or H7. And that is a positive on the PCR. The discrepancy here is due to the bandwidth at the National Veterinary Services Laboratory, they have a cutoff on the PCR, and that's the CT value. So that's the amount of virus in the sample. So if we have an H5 detection, let's say in Colorado, and the CT is 36, which is just a little bit above the threshold, they're not going to get a whole genome sequence out of that because there's not enough virus to do the sequencing. So they screen the samples. Once we know it's H5, they'll do a representative set of samples that they know they're going to have good luck at sequencing, and they sequence those. So not every single wild bird sample goes on for whole genome sequencing. We get a representative set and then know that those are B3.13. Those go in the database and GenBank and compared to the rest of the samples. So don't interpret that as in Colorado, we had 3 that were B3.13 and I guess it would be 67 that were something else. We just only got sequence back on those 3 and all of those were B3.13. So some of it is simply bandwidth for the laboratories that every single, I mean, we're talking about 10,000 samples from the wild bird side, however many thousands of samples from the poultry side, they can't sequence everything.
All right, well, it looks like we have come to the end of our questions and we're just right at the top of the hour. So very interesting and timely topic. And I definitely want to thank Dr. Lennox for her time and providing the knowledge and experience she's had over the last few years on this. I'm going to turn it over now to Dr. Stephen Castle, who is the chair of our Continuing Education Committees, for some final comments.
Yes, thank you, Dr. Toring. And I want to say a personal thank you to Dr. Lennox as well. I know that you're very busy. I'm working on the other side, the Nuln side, so we are getting all those samples sent to our labs across the country. So I know exactly how busy you are on your end. Great presentation, like I said, very timely. I also wanted to put a plug in. You may have seen an email that just came out. We put one out a couple of weeks ago. The AVMA liaisons have requested to see if anybody wants to submit a proposal to speak at the AVMA conference coming up in July of 2025. It's going to be in Washington, D.C. So please take a look at that email if you have the opportunity or you have some information that you'd like to share through the One Health program at the AVMA. That's a great opportunity. And then also, if there are other topics of interest that you have for continuing education, we've got a couple of speakers that are lined up through the rest of this year and into the winter time. But we're always looking for other speakers like this and great conversations, great topics that are very timely. So with that, I think that is all, and we'll say have a good evening. And again, thank you, Dr. Lennox, for the great presentation.
You're welcome. Thanks for having me. Appreciate it. Take care, everyone.